Today I'm bringing my research that I'm doing my master's there in Brazil. I'm studying the Laetiporus genus, but I'm specifically talking about the sulfur shelf or the chicken of the woods, the orange one from the genus. So let's get started. But first, I want to pay this respect for this guy. He he has the the most important thing that happened to me to get in this path of mycology. I was a journalist, a depressed journalist. I hated my job, and it was like doesn't want to wake up to go to work. And I got this video and this quote from Gary, and this put me on the mycology thing. So I want to thank you very much, Gary, wherever you are. And thank you, Brit and Art, because I couldn't met him in Chile, but I met him through his friends and his family, and it was like the perfect trip. And I I never met him in person, but I think I met him through you. Thank you. And then, we think in Brazil, I guess we think in Rio de Janeiro, beach, soccer, football for the English, and then but we don't have this big country, and we have a lot of things there. Not only Rio de Janeiro. Some can think about the Amazon forest when you have these big trees. There's these big roots, and the kids like to play on those roots. But it's not only Amazonia too. In Brazil, we have this diverse biodiversity, big biodiversity, and a lot of sites that we can go. The Amazon, I think, is the most known from Brazil. But we have this semi-desert, the Caatinga. We have a savanna called Cerrado, the Atlantic forest that where I live, and it's like it's a tropical forest, but you can see a lot of things like mangroves and other places, other forests that is very complex and diverse forest. We have grasslands called Pampa next to Argentina, and we have these wetlands, the Pantanal. It's like a beautiful place to go to. So we have like a lot of fungi there. We can find like big fungi, little ones, entomopathogen fungi, jelly fungi, and Ascomycetes, everything. We got this big biodiversity that is, we have to study more and look for some more. So we have these big ones here, the Macrolepiota, and a lot of fungi that are not described for science, and we have to study them. So we have a lot of things there to be known. And we have this, our lab in our university is a public university that is a good place to go. And when I abandoned journalism, it was that this opportunity to study in a public school, not paying for my studies. So, and I got lucky because in the city I live, we have this university and we have a mycology lab there. And it's some kind of difficult to find a mycology lab in a university in the world. Mostly in Brazil, we got this university with this mycology lab that working with taxonomy and systematics. So it's a very, very nice place to be. And in the first class, I was like running to this lab to know more about fungi with them. And inside this lab, we have this project, and this project, you can like Google it and look for what we are working. And not only in Brazil, we have this group with some Latin American mycologists doing research in taxonomy, biodiversity. So you can find us looking for mine fungi. In this work with this lab, we work a lot. We sought with Juliana about education and children, do a lot of these expositions for them, and they love to see fungi, and giving them the opportunity to see fungi and to learn about them, and creates this curiosity in them. So they start to. A little about my story with the mycology. When I entered the lab, I started with this coral fungi. It's called Pterula, And it's related with these ants. These ants are like farming ants. You know, they cultivate the mycelia to eat. And it's an ancient group of ants that cultivate some of species that inside this genus Pterola. So I started with these edible mushrooms for ants. And then I got a jump to the edible mushrooms for humans. And I started to look for edible mushrooms in Atlantic forest. In the first year that I was in the lab in 2011, I started to find this Laetiporus sulphurus, the chicken of the woods there. That makes me feel like, oh, I want to study these big, bright orange things. And it's like, since then, I'm studying these species. Other place that we are studying with our lab is this cloud forest. We call nano cloud forest because they are little forests. They are not a big forest. They are like in this place with those big canyons with this grassland on the top, and you got these canyons that have like 1,000 meters high, those canyons. So in some places you can find this beautiful cloud forest, and it is a little cloud forest. And we find this araucaria. It's a kind of conifer, and there's a different conifer you can see there, and it's a beautiful tree. I love those araucaria trees. In this particular place, we find like some very interesting fungi. Mostly we work with polypores, but we have this guy who is working with the cordyceps, and there are like new species being described there, and bright and soft, big ones. And one of the species where we find there in that place is a different Laetiporus. We have more than we know. We thought that we had this Laetiporus sulphurus only in Brazil, and then we started to study them and started to find more in places that never been registered. So let's talk about some with the chicken of the woods. My first time reading about the chicken of the woods was in the David Aurora book, Mushrooms Demystify, when they list the 4 proofs for the newbie in foraging for edible mushrooms, that you have these 4 species that if you pay attention, you don't commit a mistake and get poisoned. The Morchella ones, the Morels, the Calvatia, Cantarellus, and our chicken of the woods, the Laetiporus. What we can see, what are the characteristics that we have to Laetiporus to identify them? They are like big basidium, soft basidium when they are fresh. They become brittle or chalky when they are dried. They have this spore print white, and it has this typical aroma that I call it Laetiporic, because it's particular for them, and it's nice. I will show you new species that we found that we found in Brazil. It doesn't look like a Laetiporus, but because of the aroma, we got this hint and started to study them, and it's just like a related species with Laetiporus sulphurus. One other thing that I most like that in them is they are the wood decomposer, and they are brown rot fungi. That means that they digest the cellulose and let the lignin on the forest, like is a manner of sequestrate carbon in forests. So it's a very good in environment that we got this CO2 in the atmosphere. So fixating this carbon on the forest and the forest floor, making the soil, I think is the most important role that they have in the forest. Another characteristic, that's the classical characteristics, is doing microscopy, and you see 2 types of hypha. This branched hypha without septum, and the generative hypha with simple septa without a clamp connection. And then this is the classical Laetiporus system of cells. And the spores are like ellipsoid and broad ellipsoid. Yeah, that's it. You can, like, with those characteristics, you can identify them pretty easy. Laetiporus sulphurus looks like they have this worldwide distribution, because you have these popular names, these common names all around the world. You can see that in Japan and in Europe and in Portugal and in South America, and Spanish to Latin America. This gave us the perspective that they are everywhere. Besides that, they are know about the edibility. They have a lot of compounds that are being studied. Here I got a big list of them. Some of antioxidant compounds that are good in anti-tumoral activity, anti-inflammatory. They can use it against diabetes too, control of cholesterol. But we have some cases of intoxication with Laetiporus too. Sometimes described as hallucination, dizziness, and disorientation. So we have to be careful sometimes to eat them. So more than 60 nutraceutical compounds they have and are being known about Laetiporus. What's the problem of Laetiporus and why I'm studying them? Till the 90s, we have this one name only for these big yellow ones, and the Laetiporus sulphurus for the chicken of the woods. But they have showed this big variety of colors and shapes. And you can see this bright yellow one, and this was another is white pored. But people always say that it was, oh no, it's only a difference. The difference that from the shape and color is that one grows in this place and another grows in another place. So they are different in that way, but they are the same species. People started to think they are too different. They are like in different regions. Is that a real one species? And then in 1998, we got this study that identified that is a complex of species hiding in this shape, in these colors, with the name of Laetiporus sulphurus. They did this mating with the cultures of them, the monosporic cultures of them, and discovered that they do not mate when they are like different shapes or from different regions, and they tested the DNA and saw that this format, it was like hiding a complex of species, like the cryptid species that we say in biology, because of the geographic distribution. The tree hosts too are different from them, and this leads to a reproductive isolation that leads to a speciation case. Here we saw this phylogeny of most of the polypores here. We have the polypores here, and we have some Basidiomycota here, and we saw the Laetiporus clade here, showing us the time, the most ancient, that we saw this in the talk of Michael Wood about how we can see phylograms, and we see the ancient species or the ancient specimens for Laetiporus is a recent ancient one in its history of evolution from this polypores. So we are seeing these species like evolving right now, and this is probably is the cause that we saw that they are similar, but they are different species right now. Now I'm showing you the new species that are being described in the world. We are seeing these different shapes and colors that they have and the different distribution that they have. This first one is from China. It's the Laetiporus xylochaensis. They have this more yellow to reddish basidium, the fruiting body of them, and they were described in 2014. We have this one from Caribe. It's a more orange one, and it grows in some different hosts too, in a more tropical area. It was described in 2012. The Laetiporus incinato, the white-pored one, that formed these rosettes, and it has these white pores. One of the species described in the time that they were trying to test, that is a different one for another from Laetiporus collected here in the United States. We got this Laetiporus coniferocula. He's more like bright orange and yellow, and is common in conifers, most common in the western from North America. We have this Laetiporus clemioporus, is another one species described for China. In its boreal areas and temperates more cold than the other, the Chinensis. And in China, especially, they are like publishing a lot of new species of Laetiporus, doing a lot of things with DNA. That one, the Laetiporus gilbertsoni, they are almost associated with eucalyptus, but they can grow on oaks too. They are most common in more hot areas. Here in the United States, I think it's more in Florida. You can find some gilbertsonia, in California too. But in South America, they are widespread in Argentina, Paraguay, Brazil. We can find them, and we can find that in the Central America too. That one, the Laetiporus huoriniensis, they grow in the eastern north of North America and in the Great Lake areas. They look like the Laetiporus sulphurus, but they have this different host. They grow on hemlocks, different from the Laetiporus sulphurus. This one is the most recent species described of Laetiporus sulphurus from Caribe. He has this different fruiting body with some lobes in the margin. Unfortunately, the host was not identified, and it's really important to identify the host for the Laetiporus to us to know the history. Another species that was described for China, it has this smaller fruiting bodies. They are more brown in the margin, so it has this clay buff color. We have the Laetiporus montanus. It looks like the Laetiporus cincinnatus, but they are more Asia and the Central Europe too. They had this rosette shape and has more whitish, pale yellow pores. The classical, the Laetiporus sulphurus, still calling the Laetiporus sulphurus for different places, but perhaps the location of Laetiporus sulphurus is only in Europe. It was described from France, so we have to do more collection and do more DNA tests to see if the other Laetiporus sulphurus that has this bright yellow and orange fruiting body, they are the same one as from New Europe. Probably not. And this very strange Laetiporus is Laetiporus versisporus. They form these pores almost all of the fruiting body. They doesn't have like sterile upper face like the others, like looks like shelves or rosettes. We have another Chinese Laetiporus. This Xinjiangiensis. He's more pale and more salmon. Looks like the Laetiporus gilbertsoni. And we have the last one, the Zonatus, included is from China too. And it has this clay buff, and it looks like an old one, but he's always like that strange and more whitish fruiting body. So we have these 3 more species of Laetiporus. We got this register in literature, in scientific literature, but we don't have recent collections from them, and they are in like great places to study, like Mauritius Island in Africa, and both in the Pacific. So we got to go there and try to collect and test them with the new discoveries in Laetiporus. What we have now about Laetiporus in the world, we have like they spread all over the world. We have different species. Then we have 7 species in Asia described right now, most of them in China, but we have species in North and South Korea and in Japan too. We have these 2 species for Europe, Laetiporus montanus and Laetiporus sulphurus for now. And we have 7 species for the Americas. Most of the species described for the North America, and some species that we have to describe or we have like to resolve some problems that has the same name for South America and North America that we are studying right now. We have these 3 points that we have this new species to be described. They are being described right now, and in a couple of years we have these new species known by science in Hawaii, in Argentina, and then in South America. And in this study of global distribution of Laetiporus, they thought that could be the origins of Laetiporus in North America or in China. But if you see the map, we don't have collections in Australia, we don't have collections in the island in Pacific, we don't have collections here in Africa, and probably we will encounter some Laetiporus here, and this point of origins can be different, and we have a lot to do with this taxon. So we have another species of Laetiporus that are totally different from this. We have this Laetiporus persicino, is a more pinkish one. It's always on the roots of trees. We have this like crusty, recipienate fruiting body, Laetiporus squalidus. It was a collection from Brazil, only one collection, and it was when it was described. And in my work, we found another one. So we are studying them and looking for some features and seeing what we can do about the taxonomy of these species. We have those two ones from Europe and this one from South America, from Argentina. They are really different and very rare. We have to collect more than and study them doing the molecular biology tests to know how they are related and how they are related with the Laetiporus sulphurus. Now I'm showing some of the collections that we are doing in Brazil. This one is the Laetiporus sudamericanus. It was the Laetiporus gilbertsonii. We are like studying them and proposing this new name for those collections from Brazil, because in 2011, when I started with Laetiporus, I always find the Laetiporus on eucalyptus. And then 2 or 3 years, we started to find that in those native plants, they call it Pleuroma granulosa, and it's like widespread in a lot of cities in Brazil. And we started to find that in different regions of Brazil on this tree. So we tested the molecular biology, and it's showing some kind of different from Laetiporus gilbertsonii. They are closely related, but perhaps they are like adapting themselves to the climate in Brazil and the South America climate in a process of speciation. We are seeing that right now. We have this different Laetiporus from Chile. They have this shape that is very odd, and sometimes it looks like the cincinnatus, but it's totally different from cincinnatus. Probably it's a new species and a very different one, and we have it there in a different host, never described it for Laetiporus too. We have these big ones that was being like identified as Laetiporus persicinus there, but we started to do microscopy, and they are totally different from Laetiporus persicinus. Some of people were calling them Phaeolus chevenitsa because they have like this dimetic hyphal system, and Phaeolus has a monomitic hyphal system. So we started to look for the literature, the ancient literature, and it has a name, but it has a name that was synonymized inside Laetiporus persicinus. So we have to do a lot of work in taxonomy and this historical registers from this taxon, and try to make a new combination of name for this Polypores taupi fungus. And we have, for the last our collection, we have this crusty look-like fungi. That one, it was this kind of strange collection that I made, that I was looking for another fungi, and I was, oh, what is this? And when I cut, that the smells of Laetiporus came, and I saw, oh, this looked like a Laetiporus smell. And when I go to the microscopy, I saw the same patterns of binding hypha without septa and other generative hypha with simple septa, and I got to this described species that was one only collection in there in Brazil. That was the second collection there. But I started to saw another features in microscopy that is they are not from Laetiporus, and now we are doing this new combination for a new genus for this taxa. What we have right now with Laetiporus, we have a lot of work to do. I started this crowdfunding because sometimes doing the science is difficult and expensive. And I'm doing too this communication, science communication. I have this channel on YouTube, an Instagram, and a Facebook page called Cogumelogia in Portuguese, trying to show to the people how important is to know your mushrooms, to go there and collect them, identify them, learn about how we can know the mushrooms that are around us, because that's the way that I started with mushrooms. I wanted to know which mushrooms we have, and I stopped with the Laetiporus, and they are so interesting. We have this species that had one name and turned out to be dozens of species, perhaps like we have 20 species in a couple of years with this shape. And then we are like changing everything inside the Laetiporus and saying now Laetiporus genus is only for this bright yellow ones, and we have another genus inside them. And what do you know about the genus Laetiporus could become like a family inside the polypores fungi. So it's so important to continuously make science, and then is the end. Thank you.